Awesome. Thank you so much, John. Thank you for the introduction. And good morning, everyone. I'm going to be presenting today just a little part of my PhD research. I've been working, I'm a PhD student in the Horticultural Sciences Department at the University of Florida. And I've been studying this little world of grafted blueberries. But to start talking about it, I need to start talking about blueberries, right? And how they behave and how they like to be grown. Blueberries, they have a few specific requirements to grow. They are very picky to the environment they are growing. So they always prefer a soil that has a higher organic matter content and a lower pH, which is not the case from Florida. So here in Florida, the production system for blueberries needs to add the organic matter to the sand, mainly sandy soils here with no organic matter at all. And these soils are also calcareous, so they have a higher pH, which is not ideal for blueberries. Blueberries are acid-loving plants, so they need like the 5.5, 5.8 pH to be growing at their best. They are like the swampy wetland plant. They really need the moisture. That's also a reason for the pine bark to hold this moisture better than just the sand. And of course, fertilization, we use a bunch of fertilizers, and preferred form of uptake for blueberries is ammonium, not nitrate. One very important part of the morphology of the blueberries is the roots. So all of this is done because of this root system that the blueberry plants have. As we can see in this graph, in these three different cultivars, uh, the roots of blueberries are usually located in the top 35-ish centimeters of the soil. They don't grow very deep. So the deeper you go into the soil, the less roots you find, and usually no roots at all, and around 1 meter deep. So what is our big problem here? Is that irrigation and all of this costs a lot of money. It's not just the water that goes into the plant. It's not just the water that the plant needs every day to grow. Is also the entire infrastructure that the grower needs to put when he's establishing the plant. There's the energy cost for the system to be running all day, every day, and the maintenance, because when or not, the systems are very prone to failing. And the last thing you want is a row of plants that died out of nowhere because one of the irrigation lines got unplugged or something like that. So what is shocking is that basically half of the total expenses that a grower has to establish a blueberry field goes into the irrigation and into the pine bark. All of this to grow blueberries, right? So we're trying to reduce costs a little bit. And a few years ago, my advisor, Dr. Williamson, and a few other collaborators started the research and started questioning if rootstocks could help. And why is that? Other crops, other fruit crops like apples that we eat every day, peaches, oranges here in Florida, every orange tree is basically in Florida is grafted. And these plants are usually grafted to enhance plant resilience to challenging environments, to optimize plant nutrient uptake, can be to reduce to work with the plant size or plant architecture, as is the case for the apples, and of course to improve the fruit quality and the yield, hopefully. But one thing that these plants have in common that blueberries don't is that all these plants are trees. and blueberry is a bush. So when you look at a blueberry, doesn't come right away that you would graft a plant like this because it doesn't have a trunk, doesn't have that monopodial structure that would be necessary to graft a blueberry, right? However, we know we have the sparkleberry. The sparkleberry is not a blueberry, of course it's an edible fruit. It just doesn't taste very good. Birds love it. I don't. It's called Vaccinia arboreum. Is a very close relative to blueberries and is a native plant here from the southeast. So that means that is a plant that is better adapted to the soils that we have here, so can tolerate a more neutral soil pH, doesn't need the um, acid environment like the blueberries. These plants have been observed to have a deeper root system than the shallow one of blueberries, and I'll show better pictures later of that. And one of the very important things here is that we can transform that blueberry bush in a blueberry tree. Again, that monopodial trunk that the blueberry plant don't have, we can find in the sparkleberry. And that's what makes this plant very suitable for grafting. And another place that we can reduce a lot of costs in the blueberry production is with harvesting. Today, basically, most of the harvest is done manually. So it's very expensive. So a lot of work, a lot of people, a lot of actually handwork, manual labor that go into it. Because the way that the machine works to harvest doesn't work very well for the bushy architecture. So with a single trunk, that can also be improved. But these are just some of the benefits. Today we're gonna focus on the roots. To start, I started with one experiment in the past that I was just comparing some grafted plants that were grafted with the cultivar Sentinel with unrooted plants from the same cultivar in these soil columns, as we see in the picture. These soil columns were like almost a meter tall. They had a root of observation tube inside, and we grew these plants for 18 months. So here's how they look like inside. Bottom part is the sand to represent how Florida is. So Florida sand in the bottom, the pine bark layer on top. And we were looking at how these roots are growing. To do that, we had an acrylic root tube inside each of the container. And with a mini rhizotron, we were scanning these plants, these root systems, every month to see how much they were growing. So we could have like this clear picture of how deep the roots were growing. At the end of it, we decided to destructively harvest the plants to see how much roots were growing in the sand and how much of the roots were growing in the pine bark for each of the treatments, for the grafted and for the unrooted plants. Well, we learned that basically unrooted plants had more roots in total, a larger amount of roots, but that doesn't necessarily mean a lot. So we needed to look a little further, right? We want to know where these roots are. So what matters in this part is that while the unrooted plants that we can see on the right bar here, while the unrooted plants put 84% of the root system only in the pine bark, the grafted plants put 55% on the pine bark, but 44 in the sand. So this means that the roots from the grafted plants were growing more in the sand than the roots from the unrooted plants. But as I was saying, it's not just where the roots are that matters, or just there's more to it. So with those images, we could see how the health of the root was looking. On the unrooted plants, we could see very fine roots, those roots that look like literally a hair. And also these darker roots that we see here representing root depth. So the roots of the blueberry plants that were reaching the sand, so that were growing deeper, were not necessarily were not functional. And when we look at the grafted plant with the sparkleberry root, we could find structural roots growing down in the sand. So down in those 44 to 66 centimeters, we had healthy and structural roots, not just those fine roots of blueberries. And here, this is another picture just for reference of how this looks, how these roots look like in the environment. This is another cultivar, 6 years old. Both plants were. And what I'm talking about here is the size of the roots, how shallow it is a massive fibrous root. It looks like a bunch of hair. While the grafted plant, it's a totally different root system that we would expect would benefit the plant. So now that we know that the grafted plants have indeed this deeper root system, and when we want to know what can this root system do for the plants. So one of the experiments that I've done is about water deficit stress. So for this one, we compared blueberry plants, again, unrooted with their normal roots, to grafted blueberry plants. The Sparkleberry Rootstock with the same blueberry cultivar as a scion. We used this planting that was a little older, plants were very well established. And this experiment happened throughout 10 days, basically, and it had 3 phases. The first days of the experiment were the pre-drought or the acclimation period. That's the part where we were just looking at the baseline results. So just the baseline for how the plants are doing normally without any water deficit. Then we follow by a drought period of 9 days. And finally, we turn this irrigation back on in the end of it to get the recovery results. All of this structure was done to prevent any rain to affect the experiment. Again, this is water deficit. We cut irrigation. We didn't want any rainwater or anything to get into these plants. So we had this plastic cover all over the field, this row, and a Tyvek layer on the ground to prevent any water that could get on the floor to actually reach the roots. So once again, we wanted to confirm where these roots were. We did a little sampling of using basically a PVC tube and getting the roots that were 80 centimeters down the soil. So we collected the soil and the roots present in this tube. And we separated in 4 different depths. So 0 to 20 centimeters, 20 to 40, 40 to 60, 60 to 80. Basically, there was not much differences, even though it looks like unrooted plants had much more roots in length and in dry weight. What we need to know is where the roots were, like before, right? And as we expected, the roots of these plants were just like what we saw in the greenhouse experiment. So while the unrooted plant had 60% of the roots in the top 20 centimeters of the soil, the grafted plant had basically half of the root system, or more than half of the root system in the top 40 centimeters. And even like going from 60 to 80 centimeters down, we got almost 10% of the roots found on that part of the soil. Again, just confirming our theory or our idea that these plants are actually deeper rooted, and that this might be the reason why you're gonna see the results. So anyways, so if these roots are actually doing something for the plants, we decided to look into a few physiological parameters. Today I'm gonna talk a little bit about the gas exchange, where I used Cyrus 3 and Cyrus 4 for some measurements, and water potential, that I had a psychrometer logging in real time, the stem water potential of the plants. For the gas exchange, we decided to work with 3 specific time points, one before the drought, one on the peak drought, and one after the one on rehydration, so after the plants got the water back. And another important thing is that this assimilation values are gonna be a little low, and gas exchange too, and stomatoconductance too, because the measurements had to be taken at 8 a.m. We found that out doing a preliminary experiment and realizing that the blueberry plants tend to close their stomata in the middle of the day, because to avoid any extreme transpiration unnecessary and the wilting. So what we could see is that assimilation, so photosynthesis, was definitely affected by drought, and that the unrooted plants reduced their photosynthesis much more than the grafted plants did. And same for the stomatoconductance. So while the grafted plants were basically on the same level, doing their thing, basically, the unrooted plants were feeling the droughts and reducing their stomatoconductance, lowering their photosynthesis, consequently lowering growth and everything that comes with it. And surprisingly, with the stem water potential results, we were doing, again, the 3 phases. So acclimation, everything was normal. This is measuring how much water there is inside the plant, or how stressed the plant is. This flat line here means everything is good. When you go to the drought period, when we stopped irrigation, the top graph is unrooted plant, the bottom graph is grafted plant. The unrooted plant, and this is just a one plant sample to understand the data, that the unrooted plants started decreasing their stem water potential right away, right after we cut irrigation. And while the grafted plants didn't even feel the drought. So they were not affected at all by the drought. This is our results for the pre-dawn measurement. So right before sunrise, that's the point of maximum hydration of the plant, the point where everything should be a flat line. But we were seeing that we saw that the unrooted plants were decreasing basically 0.2 megapascals a day in their water potential, while the grafted plants, as we can clearly see on this graph, they didn't feel the drought. They didn't realize that they were lacking water. We actually had to stopped the experiment a little earlier than we wanted to, because the unrooted plants started showing symptoms that we didn't want to see. We didn't want them to get to the point of extreme stress that they wouldn't recover. We still wanted these plants to be alive for further research. So we ended up the experiment a little earlier. However, this year, things got a little crazy. What we didn't want to happen happened. The irrigation line didn't work for a few weeks or a few days. We don't know exactly how long, but this happened. And when you look at this picture, I know it's a little confusing, but these are the same plants that I used in the experiment. After I really don't know how long it took, but after a few days without water, the unrooted plant, this dead plant here, just died. And the neighbor plants, the green ones, are both grafted plants. And they are doing perfectly fine. They didn't show any symptoms. They were not yellowing, they were not stressed. And again, we lost the unrooted plants from that row from this experiment because of water. So basically, that's the summary of my research is that yes, grafting will increase the rooting depth of blueberries. Yes, that can reduce the stress during the day and the stress during a period of drought. And also these plants can, even during a stress period, maintain a high photosynthetic rate. This research has a long way to go, but we also have done a bunch more about this grafted blueberry world. Just some of our findings that we know for now is that these plants can actually do very good and produce high quality fruit with less pine bark than the unrooted plants need. And in this same environment, they can produce higher yields. As we just talked, they can be more resilient to environment and climate change, produce fruits with better fruit quality, more data that I can share the other research we have that is published, comparing these plants and showing how the fruit quality improved with grafting. And of course, longevity, something I haven't mentioned before, but here in our research site, since this research started 15 years ago, there's one field that used to be grafted and unrooted plants. And after, I don't know how many years, I think like 6 or 7 years, all the unrooted plants started decaying. And today that field doesn't have any of the unrooted plants, but all the plants that were grafted, again, 15 years ago, are still there. So it's very promising for longevity too. And we're still on the way to working with and figuring out if we can reduce the harvest labor, reduce the use of fertilizers, and increase profit by using grafting for blueberry production. None of this would get done without a team, so I need to bring my acknowledgements to this huge team of support that I have around me. And of course, the funding agencies that have been supporting and funding this research. And with that, thank you so much. I will take any questions. If you want to follow the QR code, it's going to take you to more information, all the papers, all the research that has been done, some podcast episode. We have a video on how to graft, anything. We have a bunch of resources about grafting, and we're trying to spread the word and make sure that this actually can help the growers with this, changing climate and everything we're going through. But yeah, I will take any questions.